Welcome. I'm Liz Taylor, ocean explorer, naturalist, and entrepreneur.
I'm Sylvia Earle. I'm an oceanographer, National Geographic explorer, founder of Mission Blue, and I'm an ocean elder, like our guest today, Rita Colwell. Thank you, ocean elders, for making this program possible.
Absolutely. And this is our show, Dive In, where we host informal and open conversations with the ocean community on topics of wonder and interest. We're really glad to be back and hope everyone has made some time to get outdoors. You can put questions in the Q&A box, and we will get to as many as we can towards the end of the program. And before we get started today, let's remind everyone that the world
Is
blue.
blue.
Very good.
So in a previous episode, we talked about sea star wasting disease and the discovery of one of the root causes as a Vibrio bacteria. And today, we are so excited to have Dr. Rita Colwell here with us, a world-renowned scientist who discovered the bacterium that causes cholera, among many others. And among her many other distinctions, she served as the 11th director of the National Science Foundation and has been awarded 63 honorary degrees
And counting.
and counting. 63, come on. She served as the president of the University of Maryland's Biotechnology Institute and professor of microbiology and biotechnology at the University of Maryland. And as Sylvia said, she is also an ocean elder. Welcome, Rita.
Wait.
Wait.
Book.
You always have to come up with a book. It's excellent.
This is really.
Thank you very much. It's a pleasure to be here. It's great fun. Anytime with Sylvia Earle is an honor anyway, so
With this
glad
one.
to be here.
Scoot over here. Get in this frame. No other way. There we go. So you're going to share your own slides today. So
Instead of somebody else.
instead of me fighting with it, which is marvelous.
Okay.
You know,
So-
we always tell people to, they shouldn't sweat the small stuff, but I think you're going to tell us that we really should be sweating the small stuff.
Yeah.
Indeed. People haven't really thought about microorganisms, bacteria and viruses in the ocean as a big deal. But we're finding that it is now because people in Florida, where the water temperature got as high as 100 degrees, and when the hurricanes sweep through, they've been getting sick with Vibrio vulnificus, and a lot of them have died. It has a very high infection rate and a fatality of about 50%. That's tough.
Very significant. I mean, most people feel like, oh, I've just got a great day on the beach. I'm going to go out and kick around in the surf, try to cool off even if the water's hot. But, there's been some cases where people just go to the beach and they end up in hospital.
Well, last October, the beaches in Providence, Rhode Island were closed because a couple of people died from Vibrio vulnificus infections. So I'd kind of like to tell you about the Vibrios and sort of the background that goes back about 60 years when I started as a graduate student at the University of Washington and found that the most common bacteria associated with fish and shellfish and seaweed and so forth were Vibrio species. They were easy to isolate and present. And at that time, there were maybe half a dozen species, but we didn't consider them as pathogens. In fact, I think I'll share the slides and see if I explain. See, what was interesting is that it was understood that Vibrio cholerae, that's the red ones, was a dangerous pathogen. But the other species, Fluvialis and Alginolyticus, weren't even described when I was a graduate student. They weren't described till about I think Parahemolyticus was described by a Japanese professor in 1950. But see if I can get this to.
Well, I remember the marine microbiology text by Claude Zouvel, and it was thought that microbes, well, bacteria in the ocean were kind of rare compared to what was known about the microbial world on the land, and we had a habit of calling most of them germs.
germs.
That's right, germs. Well, Vibrios, it turns out over the years, since I was a graduate student way back when dinosaurs were roaming the earth, they're very widely associated with fish, um, plankton, particularly plankton. They're a food for shellfish and um, just part of the natural environment.
Yeah.
However, we found out that these Vibrios are autochthonous. That's a fancy word, meaning they're naturally occurring in any aquatic environment. And I want to emphasize this means lakes out in the western United States or the Chesapeake Bay, um, Potomac River, as well as the beaches, beach areas, and the ocean. So they're aquatic microorganisms. And now there's over 100 species, close to 200 species have been described. That's in the last 50 or 60 years.
Wow.
And the Centers for Disease Control, CDC, recognizes any species in the family Vibrionaceae, any Vibrio is a reportable pathogen. So these are naturally occurring, but you see, they're present in very, very low numbers. But when they proliferate, like Vibrio cholerae, if you don't take care of having safe water to drink, then if you have 1 million per teaspoon, you come down with this really, really serious, life-threatening diarrhea, vomiting, and so forth.
Right.
Now, the discovery we made, this was in the Chesapeake Bay, way back in 1975. That's a long time ago, 50 years ago. We discovered that Vibrios, including Vibrio cholerae, were present on copepods. And
That's wild because copepods are just like everywhere, right? And they're just
absolutely.
And they're so tiny that most people don't even know they exist.
no. We had a group that was doing the film The Racing with Copepods, and kids were like discovering copepods for the first time, seeing them under a microscope and how quick they are, right? They're really kind of a fast little animal
In a jerky sort of
in a
way.
jerky way.
Yeah, well, they're fascinating.
Yeah.
Well, the argument was when being a woman scientist, and what do women scientists know, right, Sylvia? In those days,
Those days, yeah.
maybe not so true now, but in any case, the fact that I isolated Vibrio cholerae from the Chesapeake Bay, it was fascinating because the write-up was in the Washington Post, and even before my post copy arrived on my doorstep, I got a phone call saying that my funding was being cut off because it was giving bad publicity for the Chesapeake Bay.
Oh my God.
This was 1975. Well, these are the copepods. They're fascinating little creatures.
They are.
They mean us no harm.
Vibrios attached to the chitin. The chitin, that's the hard white shell of shellfish, crabs, and in oysters, and certainly in copepods. And it turns out that all Vibrios have enzymes that break down chitin. In other words, if we didn't have Vibrios in the Chesapeake Bay, we might have the bay paved in
Chitin. Little skeletal pieces. So do they kind of function as more as a parasite, or are they just there to kind of break down, breaking down?
They're just they fix nitrogen, some of them, and they're involved in carbon cycling, and certainly the chitin, that hard shell, is recycled. So they play an important environmental role. So we only very recently, in the last decade or so, realizing that, well, they can make us sick. Because until some of the work done on Vibrio balnificus, and we've always known that Vibrio cholerae was a pathogen. However, when I isolated Vibrio cholerae from the Chesapeake Bay, it wasn't believed that they were the same as the Vibrios that came from the guts of patients who were either dying of cholera or sick with cholera. And it was only until 1970 when one of my graduate students, Ron Citarella, an interesting guy, he never drove, he never got a license. He was from New York City and said you didn't need a car. Anyway,
I've met some people like that from New York. They never drive.
but he was a very, very good microbiologist, biochemist, and using a very primitive, because this was before all the sequencing machines were available.
Right.
He had to do a very tedious labeling the Vibrio of interest, let's say Vibrio cholerae, with a radioisotope, and then you would match it by using a very elaborate centrifugation technique. And we showed that in 1970, this was, 50, 35, 40 years ago, we showed that they were identical. But a lot of medical people, physicians, would not accept that.
Wow.
I got criticized because I was told that bacteria from the environment, the Vibrio from the environment, were not pathogenic. Well, they certainly are. It forced me to do the following experiment, which is now quite well known. If the Vibrio cholerae were associated with plankton, you could filter them out, the plankton, and you'd filter out most of the bacteria, not every single one, but you needed, from work done by others,
Right.
you needed 1 million per teaspoon, okay? But if you could reduce the numbers, then you should reduce cholera. So what we did in this study funded by the NIH, actually funded by the Nursing Institute, NIAID, decided it wasn't sophisticated enough.
Oh,
so
man.
they lateraled it over to the Nursing Institute. But thank you, Nursing Institute.
Absolutely.
Funded us to do a 3-year study where we went out to the remote villages in Bangladesh. And we had done our lab work showing that you could use sari cloth folded 4 or 5 times. And using the electron microscope, we showed that it would trap the plankton, because they're about 200 micrometers.
Right.
The bacteria are about 2 or 20. So we wouldn't get all the free bacteria, but we would get all the ones attached. And it was important for the women to rinse the sari cloth after filtering it. You'd fold it, put it over the top of the calash, which is where they collected the water for their family for the day. And then after filtering, you rinse it, and then you dry it in the sunlight, because the sunlight is essentially a disinfecting.
Yeah.
And so it was pretty clear to the women. I mean, on the right-hand side, the water is as clear as you would see from the tap here in the US. And that shows all the stuff swimming back and forth. And the women understood when we explained that that's what carried the bacteria that made them sick.
Yeah.
Well, the studies showed that we reduced cholera by 50%.
That's incredible.
Just by simple filtration. And nylon filters were being used in Africa to remove a pathogenic agent for actually not a bacterium, but a parasite from water. But the sari cloth was more effective and also didn't cost anything, because you just used old sari, folded it 4 or 5 times, and there you were. So that's what we had to go through to prove the connection with plankton and the connection with cholera and the bacteria in the environment. So that's what we did. And so we developed this model that in the spring, with sunlight, warmer water, algae bloom, the phytoplankton respond to the sunlight because they have the mechanism for harnessing sunlight for energy. And then the plankton, of course, come whipping in, feeding on the phytoplankton, gobbling it up, and then when they have essentially gorged on the phytoplankton, then that population crashes because there's not enough food to maintain them all. Then the vibrios are released into the water, and that's when people drink it. And then, obviously, with fecal shedding into the water, because in those pond systems, one corner is a latrine and the other corner is where you collect the water for the house.
Yeah, that's right.
And so that's how the cycle then proliferates. You know, we made the connection. This was way back in 1985 when Landsat was launched, and it measured chlorophyll, sea surface temperature, and sea surface height, tide. And so we had found that this relationship with phytoplankton and zooplankton meant that we could use satellite sensing and correlate it with the outbreaks of cholera. And that's exactly what we did. And it took us about 5 years. We published it 25 years ago in PNAS in 2000. And we showed this relationship. And this chart shows the direct relationship, or pretty good relationship, between the temperature of the water off the coast of the Bay of Bengal and the cases of cholera in red that were occurring. Mm-hmm. So as the water warmed over the years, the bacteria would proliferate, and taking water from the brackish area, which is what women did, to the village water supply, there was this direct correlation with temperature and with the chlorophyll, meaning the plankton populations. And so we were able to suggest that we could use satellites to predict conditions that were optimum for cholera. So here's a bacterium in the estuary, and we're measuring it by satellite. We started out at first with Landsat back in 1980, 1990, 2000, and that study we published. And then we've gone on, as the satellites have gone up more and more sophisticated, to the ones where we now can measure, not as we did with this early satellite, a mile square. We now can measure in a pond conditions that allow us to predict whether or not there'll be an outbreak of cholera. And we did this for Yemen in 2017. We took the satellite overflights, and we then calculated on our model, our computational model, where geographically there was a high risk of cholera. And so the colored slide shows that our prediction was, where it's red, is where the cholera was most likely to occur. And as you can see from the actual map and the lower, that's where the cholera did occur. And so we were asked by the UN agency to provide them with prediction maps. And we've been doing that on a regular basis ever since, including even now doing overflights in the Gaza region, using this combination of temperature, chlorophyll, computational modeling, and being able to put those conditions together and predict when it's likely there'll be a cholera outbreak. So we've done this.
This is really critically important for, for public health and just this kind of predictive forecasting. We see working on other kinds of predictive models, but this one seems like there's so much good correlation with the temperature and the populations and the water quality and sanitation. It just all kind of comes together quite nicely.
The loss of stable natural habitats too. I mean, so much has been disrupted that once might have been a safer ocean, but losing so many of the seagrasses,
mangroves, wetlands.
Yeah,
absolutely.
The natural filtration that, plus the density of population.
Just stable systems where you don't get these disrupted anomalies, which are
And now
horrible.
that's what we did with cholera. But what we're finding out is that there's other vibrios. Now they don't cause pandemics, they're not causing like we even today, 100,000 cases a year and a large number of deaths from cholera. We're in a pandemic that has spread out since 1960. In fact, during COVID, we had two pandemics, but we didn't pay much attention to the cholera pandemic. It was mostly in South America and Southeast Asia. But these other vibrios, as the climate has changed with warmer water, more severe weather, hurricanes, we're being more exposed to larger numbers because they're proliferating under these conditions that select for them.
Yeah, it's great for them, but it's not great for us.
Well,
Exactly.
you have disturbed systems because
Because they're supposed to be there cycling carbon and nitrogen.
Yeah.
Mhm.
And and you see like so frequently now, I think we saw in Florida with these big sequences of some of these very large hurricanes that came through, and then more recently in Jamaica and other areas. But there's so much area that's been paved over, so the storm surge comes much further in and really inundates areas. Plus, you have what used to just be floodwater isn't floodwater anymore. It's contaminated with all kinds of chemicals and manure and raw sewage and things like this. It must just be, like a smorgasbord for bacteria.
Absolutely. Nutrients for the bacteria. And so the two, in addition to cholera, are Vibrio parahemolyticus, which is mostly a foodborne. If you eat contaminated shrimp or clams or oysters or whatever. And then Vibrio vulnificus, which is an insidious bacterium. If you have a cut and you're swimming or you're a fisherman and you're hauling in your catch and you cut your hand and it gets infected with Vibrio, if you don't treat it, you can either lose a limb if you're lucky, and there are numbers of cases of amputations of hands and legs because of the infection.
Is that the notorious flesh-eating bacteria?
That's exactly what they're called,
Yeah.
right? And they're naturally occurring in aquatic system, but in very low numbers because they're outcompeted until the temperature goes up, runoff from storm surge, which is nutrient,
Yeah.
and it also kind of reduces the pH
Oh,
because an alkaline pH of high nutrient, alkaline pH, and an appropriate salinity, that's how we select in the laboratory. So now we're creating these conditions in the environment off the coast of Florida, off
swell.
the east coast of the US, even in Alaska. Cases of Vibrio parahemolyticus, food poisoning, have shot up.
Right. And it's called that because it does sort of paralyze the parts of the nervous system, doesn't it? Is that kind of one of the symptoms of it?
With Vibrio parahemolyticus, it's sort of how should I say, a milder form of cholera.
Ah, okay.
Less likely to be fatal. It can be, but less likely.
Mhm.
And you just sort of wish you'd die, but you don't.
we can look at the physical changes of the coastline that are disruptive, but they're also the composition of what has been for long periods of time before humans even arrived, where there was a lot of give and take, but it resulted in a kind of stability that was still there until we really learned how to, with new technologies, to find, capture, and market ocean life on a scale that has never, ever been a part of Earth's history before. One species, that would be us, disrupting and consuming so many creatures from the sea, whether it's oysters in Chesapeake Bay or New York Harbor or anywhere that humans and oysters have come in contact. We managed to wipe them out, the natural filter feeding organisms and the entire food web. I mean, I don't know, nobody knows exactly how many species no longer exist because we have so disrupted coral reefs and other areas by taking so much of the wild animals that are necessary to maintain stability, whether it's a
Right
coral
on.
or flat.
Yeah. And so when people have Sylvia always says a oyster is much more valuable alive than dead on the plate, or or
Yeah.
lemon slices, or on the half shell, but
And clams too.
and clams. But do they sort of bioaccumulate the bacteria in their systems as they
and filter
filter?
the water.
But I mean,
Right. And these are the typical infections that would occur with a Vibrio vulnificus infection.
mhm.
But notice that the cases are increasing.
Yeah.
This is from 2006 to 2019, and we have data to the present, and it continues to go up.
Maybe we
And
need to
the
bring back
different colors of the different pathogens, the different Vibrio pathogens. This is in Maryland.
Yeah, as you were saying, the role of the menhaden, because they must eat a tremendous amount of copepods, and
yeah, of course they do. They're
but
filter feeders. They
if
feed right at the bottom of the food chain. Others are middlemen. They eat the zooplankton that are, of course, consuming the phytoplankton. But in any case, we have so seriously disrupted these natural rhythms that have been maintained over gazillions of years until our numbers of, let's see, since 1980, our numbers have doubled.
wow,
Right.
And our concern,
And this is Florida.
yeah,
wow, look at that, that's stunning. Yeah,
And the the red of the Vibrio vulnificus infections. And this is just the listing of infections with Vibrio vulnificus and other Vibrios. And
incredible.
what's fascinating is that has, over the last decade or so, a couple of decades, there's been a six-fold increase in the non-foodborne cases. That means, infections of Vibrio vulnificus of the skin and invasion. And it's moving, now this is what's important. It's not that the Vibrios are, moving north, swimming their way toward Maine. It's that the environment is warming.
Hmm.
They're there in very low numbers, but because the conditions are optimum for them to multiply, they
They're all
become
about
more
it.
numerous, and it's more likely you get infected.
Yeah, because as you said, they tend to be like very widespread, but when their numbers are low, it's not really a concern for us or for our health. But when the conditions are right and they're like, woohoo, they just
Exactly.
they just go wild and really just numbers go way up, so our
but
chance of being infected is,
but isn't it? It's more than it is temperature, obviously temperature related, but the loss of the components of a healthy, stable system, clams, oysters, men,
Yes.
all the normal filter feeding creatures
Right on.
correlation that their numbers have gone down as our numbers have gone up as the Vibrios have gone up because of
The Vibrios have a free reign.
yeah. And I was reading the I thought I read that many of them can go into kind of a dormant stage, so they can kind of hang out for a while and before they become, conditions are really optimal for them to reemerge.
That's exactly what we discovered and published. And again, we weren't believed. The CDC uh, referred to them as Colwell's ghosts.
Oh my Lord.
So let me explain. As Sylvia keeps pointing out, rightfully, the bacteria are associated with copepods. Now, in the Chesapeake Bay, the copepods during the winter go into a diapause. They burrow into the sediment. They go to sleep. They go into dormancy. The bacteria are associated with them. So the bacteria go to sleep, and you can't culture them. And that's why in Bangladesh, they couldn't figure out where the Vibrios were coming from because between epidemics, you can't grow them. They're alive, but they won't grow on the usual laboratory media. And WHO published a book in 1948, edited by Dr. Politzer, where they reported that they tested cows and goats, water, and everything they could think of, and they could not isolate it from the environment between epidemics. And we showed by actually, it's kind of fun, by making the bacteria radioactive, by feeding them a radioactive amino acid, and then just plating them, putting them on a background that will detect the radioactivity that they emit when they metabolize the amino acid. They take their own picture, so to speak. And so we demonstrated that they're alive, they're not dead. And then we were able to do an experiment where they were testing a vaccine for typhoid fever. And this was at the University of Maryland Medical School. And we were allowed a few volunteers to ingest these sleeping Vibrio cholerae.
Oh, man.
And within 4 or 5 days, and it was we had eliminated genetically, Jim Caper, former student, had excised the gene for a couple of the pathogenicity traits. So what happened is after they drank some of the Vibrio, attenuated, weakened Vibrio cholerae, within 4 or 5 days, they began excreting the cholera Vibrio, which we could culture. And of course, we immediately gave them antibiotics, but we didn't want typhoid or cholera.
No.
And cholera Jane roaming around Baltimore.
No.
But we showed that they were alive, but they'd gone dormant. And that's what they do in the natural environment.
Yeah, that makes sense. I mean, I guess there's a lot of animals that sort of hibernate or go dormant, and so why wouldn't a bacteria, especially if the host is doing that, you know?
That's exactly right. So that's another funny story.
That's a great story.
And so, what are the pathogens along the coast? Well, here you can see Florida, Louisiana, Texas, Pennsylvania, and so forth. And then the color indicates, for example, the orange is Vibrio parahemolyticus. The dark green is Vibrio vulnificus. And so you can see there's a range of these Vibrio species all the way up the East Coast and also in Oregon and Washington on the West Coast. So we've been able to map these from the literature and from our own work, showing that they're widely distributed, but now that the climate has changed, the infections that they cause are going up.
Yeah, I remember in Seattle area, there was a couple of years ago, they had sort of that massive heat wave, and they had so many of the oysters and mussels and things that just died. But what happens to the Vibrio in those cases? Do they perish under very high temperatures, or they just kind of
Now
like
they end
head
up in the
out
sediment.
down to the sediment? Yeah, so they're persistent.
Yes,
Wow.
very much so.
I think increasingly it's important to realize that climate is increasingly an obvious driver of the changes, but it's also the disruption of the ecosystems that has really progressed with the advance of technology in the last 50, 60 years that has enabled us to eliminate like 90% of the sharks and a reduction of ocean wildlife overall since the 1950s by about half, which means you don't have the ocean that once existed And what it's continuing. We're continuing to add the biological factors along with the physical ones, and they're interrelated. I mean, it's a living planet.
Yes.
And so we make physical measurements of things like temperature, salinity, acidity, but they're driven by the fact that this is a living planet. It's not just rocks and water, and everything interacts with everything else. But as I heard you say, microbes rule. In what sense they govern the chemistry. They they make our existence possible. A
Yep.
few of them get out of line.
A little
But
bit
we're the
rowdy.
ones out of line.
Right on, right on. And so these infections have increased over the decade. And the other thing we have observed, especially in Chesapeake Bay, is that you would have these infections uh, uh, maybe late August, early September. But now we're finding that these infections are occurring as early as May and June
Wow,
and as long as November.
incredible.
So that's another
so
parameter.
there's that,
Evidence.
yeah, evidence and that is directly correlating to the longer periods of warmer water.
Exactly. And that's what this particular chart shows, is that spread from May to October, November.
How cold does it really have to get for them to kind of like lay low,
Subdue.
subdue?
Well, I think for the first time in a decade that this winter has
Florida's
provided
a little brisk.
sufficient low temperature, but it's roughly the normal winter that brings on a few snowstorms and some ice. But what's also related to these Vibrio infections are hurricanes.
Oh yeah.
Now, this hurricane that went through Florida, it came from the northeast and swept across to the west coast of Florida. And in this region right here, this boxed area, it's where the winds were the highest. And it stirred up the sediment in the water column, nutrients, lots of runoff. So the salinity is optimum right on the shore. And what happened is that there were 65 cases in that particular Hurricane Ian, and 11 people died
Wow.
from Vibrio vulnificus. So there's these heavy, severe windstorms, hurricanes, typhoons, create conditions as well for the proliferation of these bacteria.
You can really see it because you've got the wind kind of blowing the sediment in. Now it's mixing. There's carrying a lot of stuff
And
inward.
with the mangroves half gone,
Yeah.
the seagrass meadows turned into shopping malls. With we've made ourselves more vulnerable by chopping up what was a relatively intact natural system until about 100 years ago, mostly the last 50.
Yeah.
I mean, I've seen it. I'm
Yeah.
sure you.
Yeah, we've all seen it. And they say what 50% of the population lives within 50 miles of the coast.
Roughly,
Something like that. Yeah.
Concentration.
Concentrated, right in these areas.
And chopping up those natural barriers that were with more stable systems, land and sea. It's all connected.
Absolutely. And what's interesting is this was Hurricane Ian in 2022, the most recent hurricane. There were over 100 deaths and hundreds of cases. So
Wow.
it's getting worse.
Yeah. No, and people don't really think about it. They think, oh, my house might flood or I've got to evacuate because high wind's coming. But they're not thinking about
Dying.
they're not thinking that it's going to kill them from a bacterial infection.
Exactly.
That's just not even kind of on the radar. They're just
And these are marine bacteria. Well, they're aquatic, not just marine. They're aquatic. Vibrio cholerae can be isolated. As a matter of fact, we've been using DNA techniques, and we are picking up low numbers now of Vibrio cholerae. But we had cholera in Florida, in the Gulf, in Maryland, in New York, couple hundred years ago. And so if we have these conditions that are enriching for the family of Vibrios, then we may begin to have our own homegrown cases of cholera.
Wow.
We have a few now, but these are people who are eating oysters in the Gulf region, particularly from areas where the oysters grow big and fat, but it's because of the sewage.
Oh man. I always say you cannot treat the ocean like a supermarket and a sewer at the same time.
You are absolutely right. Absolutely. And then, there's something that is important here. We're looking at Vibrios for people, but Vibrios, other infectious diseases, coral reef infections, and so forth. So these are the conditions, the environmental parameters that are important. Extreme weather events, ocean currents, temperature, precipitation, rainfall.
Yeah.
It's not the seal level, it's the sea level. There's an extra L there, but maybe the seals might have something to do with it. I'm not sure.
Kind of like all things all at once, right?
Yeah.
Crazy.
So it's possible for other pathogens, human, animal, to be able to develop satellite prediction of conditions. And I think that is entirely possible. And during COVID, we also used what we had learned about cholera to provide predictions of COVID in the US on environmental parameters. So it's another factor is that you can use the environment that drives specific pathogens to be able to develop a predictive capacity.
No, I remember that they were always kind of looking at like the wastewater outflows and trying to predict the COVID infection levels. And it's great that it can be used for Vibrio as well.
So what I'll do now is stop sharing and
Yeah, we have a few
take any
questions.
questions if there are any.
We do have a few questions lined up here. I'm going to open them up here. Okay.
I have a question.
What? No, do what you do.
First, quick question.
Okay.
You were told that your funding was taken away during that first report. What happened?
1975.
How did you get your funding back?
Actually, that turned out to be fascinating because this was the marine fisheries that was giving us I don't know, something like $25,000 to do some analyses. They cut us off, but I was invited to a sea grant symposium and I gave a talk, and I was awarded a 2-year, $200,000 grant.
Like, how do you like me now?
So I've always been indebted to Sea Grant. It's one of NOAA's best programs.
Yeah, it's certainly.
All right.
All right. Okay. Let's see, go to the top here. Anonymous attendee. How much awareness does the medical community have over the increase in Vibrios?
They're beginning to understand, and the Centers for Disease Control, CDC, as I said, in the last few years, have made reporting Vibrio infections mandatory. They call any Vibrio.
Any of them.
There are many other species that have caused infections, but not in the number, of cholera, certainly, or of vulnificus or perihemolyticus. So there's an awareness, but what I don't think the medical community yet understands is that how the environment climate drives infectious diseases.
Mm-hmm. Yeah. It's indeed.
It's important to understand that when the water's warm, okay, it sounds like great for swimming, but hey, the bacteria like it too.
Sure they do. Yeah.
And then it becomes a serious problem.
Yeah.
You need a healthy ocean, a healthy planet to have healthy people.
Exactly.
Absolutely. It's directly related. And we're seeing it as the infections occur now up in Maine, which
Yeah.
they didn't 25 years ago.
No, not at all. So
But
next,
it also portends not so great for the lobster fishery.
yeah, I was wondering about that because I mean that's like a giant copepod.
That's right. That's
Massive
right.
Claws.
massive claws. The pinchy bits, you know.
Exactly.
Oh, okay. Our next question. In some California cities, wastewater is being recycled. Is this a concern regarding Vibrio infection increasing?
I think recycling is important, and I many years ago I wrote a letter to the Washington Post um, suggesting that we were wasting a big resource by dumping sewage into streams and rivers instead of building reactors and recycling the heavy metals, the nutrients, the pharmaceuticals, whatever. And I got a follow-up letter from some other who has implied that it's just another dumb female question or a point, saying that it was too expensive and it wasn't able to be done. Well, yeah, you have to start somewhere. So I think recycling is really important. And
Yeah.
recycling water systems, I think, is probably less likely with a very well-functioning recycling system to get an infection because I've actually did a study for one of the recycling plants in California. We did a study to look at the effluent or what was being recycled. And I've actually drunk the water straight from that recycling plant. And I'm still here. But the point I want to make is that the bacterial count is way, way lower
I would
and
think so.
it's controlled.
Yeah, because a lot of them are using like UV filters and or treatment to kind of kill bacteria as well. Kind of like the sunlight in the
For
field.
the cerecloth, yeah.
So our next one is not quite a question, but Sarah is saying that she met Dr. Primavera in the Philippines at a conference and was aware of your work. And they're looking at uh, aquaculture for shrimp, where they're cutting down the mangroves and then they're seeing like a big spike in the bacteria because the animals are so concentrated. And is that a concern, the sort of aquacultured shrimp
Yes.
that are in these ponds that where they cut down mangroves to kind of clear the natural systems to try to.
Yes. In fact, I was called in by the WHO to go to Brazil and to Ecuador back in the 80s, late 70s, early 80s, because they were losing their shrimp crop, which was at that time, at least for Ecuador, it was a major component of their national domestic product, gross, the income. And what had happened for Ecuador is a freezer ship had was full of the shrimp had arrived, I think it was in Spain, and they tested it, and one of the samples had Vibrio cholerae.
Wow.
And that they had to send the ship back to Ecuador. That's a lot of money.
Yeah, it's a lot of fuel, it's a lot of money, it's a lot of waste.
Right.
Yeah.
And they have upped their conditions significantly to produce shrimp that are safe to eat.
Well, the thing is, if you don't know, you can't care. We know so much more now that could guide our better behavior as long as people listen.
That's right, if they act.
Yeah, but
We also have tools. We have DNA, PCR,
right.
All
DNA
kinds of stuff.
sequencing, and we're getting to the point of, I think, Oxford Nanopore sequencer can be used in the field, and so we're to the point where we can do the test that when back in 1970, when we proved that the Vibrio cholerae from Chesapeake Bay was identical to the cholera that came from a patient, and we were disbelieved and ridiculed. Well, now we can do it, in a matter of hours with sequencing.
That's great. So,
But also, we're beginning to value the importance of healthy natural ecosystems, trying to replant marshes instead of digging them up
seagrass
to make
meadows.
more land.
Yeah.
It, it's not universal, but the more evidence of the correlation between our health and the health of the natural systems that we rely on, the more people will respect and care for and try to protect what remains and restore what we can.
But we can. Yeah. 100%.
Well, for example, we've had this massive sewage spill in the Potomac River because the treatment pipes burst. They hadn't been serviced, I think, since 1960.
That is so typical. We deal with infrastructure all the time, and it's just like maddening to like how long it goes without inspection or no thought of inspection. It's just crazy.
And the DC authorities have said that for the next year, people should avoid contact with the Potomac.
Unreal.
Think about the cost-benefit ratio.
Yeah. Sorry.
You are.
Caroline says, can you get flesh-eating bacteria just by swimming in the ocean off the coast of Florida?
You can if you have an open wound. If you have, let's say you stepped on a clamshell or a rock and you have an abrasion, and you're not going to die just because you get infected, but you have to take it seriously.
Yeah, you can't just sort of like shake it off. Yeah. Go see the doctor.
Exactly.
Anonymous is wondering about cases in New Orleans because there's so much cultural significance to the seafood industry there. Do we see kind of more happening in that part of the world?
I think they take it more seriously. I think not 10 years ago, it wasn't that common, but now that the risk is higher, first of all, people are paying attention. If they do have a cut and it gets infected, they don't, just not blow it off and say, I'll just get well. I'm strong and healthy.
Yeah, I'll take some
aspirin. Systemic, it doesn't matter if you're strong and healthy.
Yeah.
But also, I think sanitation, especially with the hurricanes that have gone through and devastated New Orleans, I think understanding keeping safe water and conditions that are optimum both for people and for preventing the bacterial growth. I think the awareness is important.
Yeah, yeah. And we see it even like in emergency preparedness kits. People are having more filtration methods for water or sterilization methods to just try to keep drinking water safe.
Exactly.
Lindsay is asking, could you share what women today can learn from your journey in pushing forward against so many odds?
Well, like Sylvia, I never let anything stop me. I found a way over, under, or around whatever the obstacle. And if you really like what you are doing, if you really love, as I do, science, and I know Sylvia loves her work deeply, you continue. You don't let the slings and arrows stop you. And I think what has changed that is good is that women particularly, and some men as well, don't put up with what we put up 60 years, 70 years ago. You always thought, oh, well, it's my fault. Maybe I did something wrong. Now the women, the girls call out and say, damn it, that's not appropriate. And I won't stand for it. And that is good.
So read the book about Madame Curie, because things are better now.
That's true.
They're not where they should be or where they will be eventually.
Yeah.
But the things that she put up with.
She put up with a lot.
Yeah.
In fact, they didn't want to elect her to the French Academy because she was just a servant girl to her husband, according to them.
And that's how women are
She
often
ended up with 2 Nobel Prizes.
That's right. Persistence,
in different fields,
tenacity.
physics
That's
and
right.
chemistry.
That's right. So that's right. Oh, by the way, a funny story I think worth telling is that, and I tell it in the book, I think, is that the American Society of Microbiology was founded as the Society of American Bacteriologists and then changed its name to the American Society of Microbiology. But it was founded about 1890, I think, 1880, 1890. And the first woman president was about 1935. And it
Wow.
was a woman who discovered that relapsing fever was transmitted by milk.
Wow.
And they wouldn't believe her because he said if that was real discovery, it would have been made by a man before, quote unquote.
No.
And then the next was Rebecca Lansfield, whose typing of streptococcus infections continues to this day. But she was elected president because all the men were at war. This was 1942, I think. Then the next president was a University of Washington professor, and that was like 20 years later. That would have been 1960. And then for some reason, I was elected president, and I discovered that the reason that there was this long pause between women presidents was because the nomination committee would meet in June, would select the candidates, and then would not post until August. And any write-in, if you wanted to write in and nominate someone, you had to do it by June. So when I was president, since the immediate past president serves as chair of the nominating committee, I called the women's committee and said, hey, if you don't want to wait another 20 years for a woman president, get going and get the nomination in. Well, there was a slew of women presidents right after that, after my presidency, I think probably for the next 10 years until the men said, hey, wait a minute. So
Get busy.
they alternate between men nominations and women nominations.
That's good.
So,
We got a few more questions to get through quickly. So Chuck says he's in San Diego. Has the Tijuana sewage flow changed the infection rate locally there? In Tijuana in Mexico and San Diego.
Oh, yes, that's been very interesting. Oh dear, as a colleague who has shown that there's a direct correlation because the sewage comes through, I guess, untreated into the Los Angeles area, I think, San Diego, Los Angeles. And there is a correlation because very strong treatment is needed.
Mm-hmm. Yeah, it's brutal.
At least you know.
You know.
Imagine if you.
Right.
Let's see. Brian is asking about the sea star wasting syndrome, Vibrio.
Yes.
Say about that one.
I give credit for the folks working on it. I have not done work, but it is a Vibrio species, and it goes back to the question that Sylvia had asked earlier about the relationship to the health of invertebrates and vertebrates of the ocean and of the aquatic environment. And that's just another example of the Vibrios playing a role.
Yeah.
Disrupted system.
Yeah. And the warming, so. Okay, Anonymous is asking, can a regular person test local water for cholera presence?
Yes. In fact, we're doing that now in some of the I use one of the small ponds on campus as a place to look for plankton and for the presence of the Vibrio cholerae. And there are, I think, is available commercially these strips that have the reagents that will indicate a positive. We had developed one ourselves in collaboration with a fellow who has a company where it was essentially a kind of a dipstick where
Mhm.
you could detect the presence of Vibrio cholerae. And we used it when I made that trip some 30 years ago to Ecuador,
Mhm.
because they had an outbreak of cholera, as well as problems with the shrimp.
Yeah.
And we used that to triage because at the time they didn't have a laboratory really in the hospital. They were diagnosing by symptoms. And
Wow.
so we were able to use the rapid tests where you could just take a bit of a stool and put it on the device and you could determine the presence of cholera. And we could separate typhoid from cholera, and that enhanced
Wonderful.
treatment.
That's good. That's really good. Okay, last question. Martin says it in, oh no, another one popped in. Martin says that they have a huge sewage spill going on in the New Zealand capital of Wellington, and higher temperatures. How could remote sensing assist in tracking to help predict distribution and impacts?
Well, when we were doing the overflights of Yemen, we picked up signals in a reservoir in Sudan
Wow.
and predicted that there would be cholera, and there was. So the the sensors, the satellites, of course, are exquisitely sensitive, and our computational model, in which we have all the parameters entered, allow us then to make predictions. We don't need a mile off the coast of Bangladesh anymore. We can use the ponds themselves, satellite sensing for predicting conditions. So you should be able to do the same in New Zealand.
Excellent. Okay, last one. Uh, when you say that sewage comes through to Los Angeles, do you mean it comes to the drinking water, or is it in the ocean or the groundwater?
Oh, it's in the groundwater. No, the study I did, I'm trying to remember, of the recycled water. Um, it was a mix of recycled and the regular water, the stream water coming in. And so the drinking water is safe.
That's great.
And the drinking water is safe in Washington, D.C. I drink the water from my tap. I have an
Yeah.
extra filter, but I've always had the extra filter, not just because of the spill.
Yeah, there's lots of things. Well, thank you so much. And before we close, we'd like to say thank you, Rita, and thank you to our producers, Ocean Elders, and thank you to the diving community. You keep coming back and listening into these great conversations. We're so grateful to all of you. Water does connect us all. Um, Remain
Yes,
curious, spend time in nature, and we're going to be back later this month speaking with Barbara Block
Barbara Block.
about big things that matter.
great. Well, Sylvia and Liz, it's an honor, a pleasure to be with you on screen. Look
Yeah,
forward
and
to
we
seeing you again in reality soon.
absolutely, I certainly do hope so. And remember, take care of the ocean.
As if your life depends on it because it
It
does.
does.
It does.
It does. Thank you so much, Rita. Thank you, everyone.
Cheers.
See you next time. Bye for now.
Great. Cheers.